Good afternoon, and thank you for your attention. So my name's Sandy, and I'm the CEO of Sangamo, and I want to tell you something about our work. As always, we refer you to our forward-looking statements. And Sangamo's a company that takes groundbreaking science into clinical stage programs to make a difference to patients. And I want to just take a moment to pay reference to the panel that just went by there. Patients are incredibly important to what we do. We have a remarkable woman called Christine Moberg, who's our head of patient advocacy. And Christine ensures that patients see our protocols, see our programs, they talk about it with us, they tell us what's really important to them rather than what's just a regulatory outcome. And they come and visit us. So maybe once a month or so, or once every second month, we have a patient talking to us. And they do two things. They remind us of the urgency that we have to get there because patients are waiting. And they remind us of the responsibility to take our time and do it well because patients depend on us. And in our area, once the medicine's in the patient, it doesn't come out. And therefore, you need to get it right first time. And therefore, you need to give the right medicine to the right patient at the right time. Now, Sangamo has now built a remarkable portfolio, taking our science and applying it to important clinical programs. We have our hemophilia A asset with Pfizer in phase 3, and there's 4 things in the clinic. There is our Fabry disease program, our CAR-Treg program, and with Sanofi we have sickle cell disease and beta thalassemia. But behind that are a range of programs, and I'm only showing you the ones that are at that kind of pre-IND stage, because our technology can be applied to just about any nucleotide in the genome, which means the choice that we have and the potential that the company has is quite remarkable. And maybe I help orientate you by telling you that we still have a number of important events to come in the few months till the end of the year. You know, it's nearly Christmas, it's nearly the end of the year, and we still have many things to happen at Sangamo. We will recruit the 3rd cohort of our Fabry disease protocol. We will recruit the first patient ever in a CAR-Treg program. Our friends at Pfizer will show long-term data up to 2 years at a medical meeting on hemophilia A, and they'll talk about the recruitment into their phase 3 study. And our friends at Sanofi have committed that by the end of the year, they will talk about their sickle cell results while still recruiting into their precision study and talk about their plans for sickle cell disease. And we can do that because we've underpinned Sangamo by great financing. Since I started in 2016, we've raised $1.5 billion, about half of it on the street, and $815 million on partnerships with Takeda, Sanofi, Pfizer twice, Kite, Novartis, Gilead, and Biogen. And of course the money is important, but I think you should think of it for 2 other reasons. One is, with each one of these programs, you get the best scientists and the best clinicians in a big company that really understands the disease. And from an investment point of view, you should know that each one of these companies looked at all the editing modalities. They looked at the CRISPR companies, precision, etc., and they chose Sangamo. And the diligence that goes into one of these decisions, with so much money riding on it, is really deep and goes right to the fundamentals of our technology. And that is why I feel that we really should be proud of the zinc finger technology that Sangamo owns. So let's talk about our proprietary programs for a moment. We're in phase 1/2 for Fabry disease, where we're recruiting patients that are on ERT, patients that are naive, and patients that are pseudo-naive. That means they've, at some point in the past, had ERT. And for all these studies, it's all about safety as the first thing. There is a discussion in the world about AAV and its safety at the moment, and I think it beholds all of us to take our time and do it well, and make sure that we choose the right patients, and make sure that, because this is their one decision about which AAV they choose, which medicine they choose. But we hope to be able to show efficacy as well, because the patient has to have a benefit out of this. And we'll show alpha-gal levels in the near term, and eventually we'll talk to you about withdrawal of ERT and all the other things that the patient benefits from, whether it's reduction in lyso-Gb3 and eventually in biopsies that show reduction in the tissues. Because when we look at this asset in mice, it's produced in the liver, goes into the plasma, and it clears all of the tissues. And it clears them of the lyso-Gb3 that damages the liver, damages the heart, damages the kidney, and gives the consequences that really changes the patient's life. And we think that the reason the patient will like our medicine rather than some of the others that require conditioning, require a bone marrow transplant almost in effect, is because it's a single injection of an AAV that goes to the liver and produces the enzyme. And those have been remarkably safe in the short term. We all have a responsibility to follow the patients in the long term and ensure that the benefit over the patient's lifetime continues. But we're very excited by the progress we've shown in Fabry and look forward to sharing some of that data with you. Switching gears to CAR-Tregs, since we acquired TXL, the French company, there's been 7 new CAR-Treg companies set up. It is the new CAR-T frontier of cellular therapy, and that's because you can take the cells out, edit them, and give them back to the patient. And our first trial is in renal transplant, so let me take a moment to explain that. 20% of renal transplants are not matched at the HLA-A2 locus. 20%. And that means that you find a patient that's HLA-A2 negative and is due to get a live donor HLA-A2 positive kidney. Before they get the kidney, you apheresis the patient, you purify their Tregs, and you add a CAR to it that will take the Treg to anywhere that is HLA-A2 positive. It's like a targeting GPS system. The patient then gets the kidney, which is the only place in their body that's HLA-A2 positive. And some period afterwards, because we want to let the kidney bed in and the transplant to heal, you give them back their cells. And our hope is that the CAR-Tregs to HLA-A2, an autologous process, will go to the kidney, will be activated in the kidney, and will release the inflammatory mediators, both cellular and cytokines, that will calm down the inflammation. Now, there's a study that was reported in The Lancet in 2020 called the ONE study, where they looked at polyclonal Tregs in this process. And they were able to show that the investigators were able to back off in the immunotherapy with polyclonal expanded Tregs, which gives us great hope for focused CAR-Tregs that will go only to the HLA-2. So we're very excited by this study, and we hope to enroll the first patient this year. And as always, it's a safety study, and it's 3 doses that will gradually increase the number of cells because, to be honest, nobody knows what the right number of cells is. And we have to be prudent and start at a low dose and gradually increase. And eventually, we hope to be able to show you that the cells have been localized, activated, and maintained. We hope to work with the investigator to gradually back off on the immunosuppression and show that the patient gets a real benefit for CAR-Treg. Now, we get 2 things out of it. We get a medicine, hopefully, to be able to treat renal transplant patients, and there's a lot of patients get renal transplants, and if you speak to them, as in the conversations we've just been hearing about, what they talk about is the operation is one thing, it's all the immunosuppression and the concurrent illness that is caused by that really is the bugbear of their life after the transplant. And we hope this is a much easier way for them. It has to be a much easier way for them. But we already have a pipeline behind the autologous HLA-A2 project. We have allogeneic HLA-A2. And so by making the cells allogeneic, it means we don't have to take it from the patient, but we can take it from either healthy donors or, most excitingly, from iPSC cells, and then make it in large batches, keep it on the shelf, and then give it to the patient just when it's ready, without any form of apheresis. And we're now expanding that, so we have programs to create CARs for MOG, for multiple sclerosis, for IL-23R, for Crohn's disease, and a variety of other CARs behind that. And then you can look on the right-hand side as our future dreams, as when we show this is successful, this could be an immunomodulatory treatment that involves very few doses, one perhaps, and allows the patients to step away from immunotherapies and allows them to step away from their disease. Let me just talk about our partnered programs. We're very proud of our partner programs. I look at them as still our children. They've just gone to college with someone else. And our partners are great partners. They believe in what we do, and they take forward the Sangamo assets with great urgency and with pride. And Pfizer has done a remarkable job during the pandemic to recruit the pre-study for the phase 3 of haemophilia A, and then to take those patients and convert them into the treatment study. They're currently guiding that they'll show results in 2022, but they will update that before the end of the year. In addition, they will show the long-term data from our phase 1/2 study, which is up to 2 years, so as everyone can get an understanding of the persistence of the effect. And our second big partnership in late phase is with Sanofi. It was originally with Biogen, it went to Bioverativ, and now it's with Sanofi. And I'm delighted it's with Sanofi, because Sanofi is a company with global reach and with a social conscience. And in the field of sickle cell disease in particular, you really need a company with that global mindset. They have guided that they've dosed 4 patients, and they'll show the results for those in a medical conference by the end of the year. They will also show the thalassemia data that Sangamo did and reported on last year, and they'll talk about their plans for this project. Switching gears to go back even further in the pipeline, I want to talk about neurological disease. When people think about zinc fingers, you think about nucleases, you think about double-stranded breaks. And yes, that's a very useful technology, but Sangamo now thinks of the zinc fingers as almost like a Swiss Army knife that has a series of different attachments. We can add repressors, as we do for tau, to turn down a gene without a double-stranded break. We can add enhancers, as we do for Sanofi in autism spectrum. We can add base editors, recombinases, with epigenetic editing. And you can see that what you end up with is a series of options to address genomic problems that if a company partners with Sangamo, they get everything in the one shop, as opposed to going 6 or 7 different CRISPR companies. And we really believe the advantage of the zinc fingers with the small size and this combination gives us a unique advantage, which is why we have a growing, burgeoning CNS platform, both with ourselves and with others. So here's the key points I want you to take away with, because this is an important year for Sangamo, an important quarter. I want you to remember that we're dedicated to delivery. We're dedicated to making sure that our clinical studies succeed and we take our medicines to the patients as quickly as possible. And you're going to hear results in the next quarter for haemophilia, for recruitment into Fabry, and for sickle cell disease. I want you to know that manufacturing is important, particularly in cell therapy. For all the other CAR-T reg companies, it's not just about editing, it's not just about publishing on Tregs, it's about understanding how to make the cell, because the process is the product. And it's essential to get that right. And we will have GMP manufacturing for AAV in California, or we have that already, and we'll have cell therapy manufacturing, GMP standard, in California at the end of this year, and in France at our Valbonne facility at the end of this year. We have catalysts coming up in haemophilia, Fabry, manufacturing, Pfizer, and Sanofi. And the things I want you to take away in the last 20 seconds is that we are a genomic medicine company with a broad capability, and that's important because it's different tools for different diseases. It's not just about applying the same thing again and again. That we are well capitalized with over 500 million in the bank, we have in-house GMP capability, And we're the genomic medicine company that's preparing for the future. We're building a medicine pipeline because that's important for the patients that you saw earlier. Thank you very much.